drug called rifampicin. Now, rifampicin is an antibiotic that is also used in treating tuberculosis, but in vaccinia virus it has a very specific effect. It precisely targets the scaffold protein of the virus in a reversible manner. So, if you have rifampicin, you make viral membranes, but they're unshaped because of the lack of the scaffold protein, which basically localizes somewhere here in the cell, so away from the membranes. And if you wash out the rifampicin, then you start synchronously making these crescent-shaped membranes. And so this is what it looks like in real life. This is a rifampicin-treated cells. There is no crescent. You have the virosomes with unstructured membranes. And then when you wash out the rifampicin, you see that now these crescents are made by the assembly of the scaffold protein on the outside. And so what we asked ourselves is, what is going on here precisely where the scaffolds start being assembled? Is it so that, is this scaffold continuous with a membrane, or is it continuous with nothing? And the data were pretty obvious. This is again the pure method without any fixing. Very obvious was that when you see the scaffold assembling after rifampicin washout, it's continuous with nothing, and what is basically not coated with a scaffold is a single membrane. So we took this really as a proof that we are not dealing with the situation here, but that basically we have one membrane with a scaffold on top. So one membrane, so basically we can start excluding this model here. But how can you make one membrane in the cytoplasm? As I told you, this is not consistent with what cell biologists think about membranes. And so we were thinking perhaps this membrane is continuous with something, but we cannot see it. We cannot see it because we have these two-dimensional sections, and in 3 dimensions it's continuous with something, but we cannot really see it. And so in order to address that question, so basically we have a problem here. You cut your slices of the orange, you have a 2-dimensional projection, and you cannot see what is going on in 3 dimensions. So in order to solve that problem, we use electron tomography. And this is the principle of electron tomography. Basically, you cut sections that are slightly thicker than usual, 300 nanometers. And then when you put it into the electron microscope, you tilt your sample. Basically, you rotate it in 2 directions. And each degree of tilt, you make a 2-dimensional image. And then you get a series of 2-dimensional images. And when you reconstruct them with a computer, you can get a 3-dimensional model of the structure. So this is the microscope, typically devoted for electron tomography. And basically, this is where you put in your sample, and this whole thing here can then basically be rotated. And when you do this, this is like a crude series. If I can get this to work, yes. A crude series as it comes out of the electron microscope. And basically, this doesn't look so satisfying because you don't see very much 3-dimensional information yet. But then, when you reconstruct it, here, using a program called IMOD, this is all computer work, you start appreciating more like the 3-dimensional structure. What you see here is a crescent, and you see these spherical immature particles, and one here that's already taken up the viral DNA here by this very large thing. So now you start getting a 3-dimensional impression. What you can do in addition with tomography, you can basically tell the computer, okay, this is my membrane, this is my scaffold, this is the viral DNA, and then you can give all of these structures a color. This is what you call rendering. And when you do rendering, this is what comes out. It's all the same series of tomograms. Basically what you see here now, in yellow is the membrane, in red is the scaffold protein, and in blue the DNA. And what becomes very obvious when you do this is that the crescent membrane is an open membrane sheet, because remember we are talking about 3 dimensions. Whereas the immature virus that has taken up the viral DNA has a closed membrane. So very nice and colorful. I have to emphasize here actually that giving colors, rendering, is a little bit subjective, because you as a person are actually telling computer what is what. So you have to be careful with this. So, open membrane sheets. And how is this made? I mean, how can we reconcile this with cell biological principles where all the membranes are closed? So, what really solved this problem here is when we started to analyze a little bit more the ends of the crescents using immunolabeling. Now, what can you do in immunolabeling? Basically, you take an antibody and you incubate your sections with this antibody, and in order to visualize the antibody, you use probes that are coupled to gold particles. Gold particles because they are electron dense and you can see them very well under the electron microscope. So just exemplified here where we have a protein that localizes to one side of the Golgi complex. It's not relevant for this study here. And when we started doing this with Vaccinia virus, with a membrane protein, an antibody to a membrane protein, we realized that the crescents are actually always in close proximity to other small membranes that don't seem to be structured. And when you analyze this in very much detail, you see that the ends of the crescents are continuous with a small membrane that is not coated with a scaffold and that seems to be turning away from the crescent. And very often these membranes actually seem to be open into these sections. They're open here, open, and here, up here. So we decided that this is important to analyze in 3 dimensions using tomography. And this is what we did here. Basically, what you can see here in 3 dimensions is a crescent that comes up, and this crescent in 3 dimensions seems to be continuous with small membranes, and when you analyze these small membranes in detail, you see that most of them are actually open. Here are the small membranes again, and you can see how they're actually continuous here with this growing crescent membrane. So perhaps in the rendering it's easier to appreciate. This is the model. Basically, we have the yellow membrane here, continuous with all of these heterogeneous membranes, and these heterogeneous membranes are all open in 3 dimensions. So, based on all of these results, we propose this quite unconventional model on the biogenesis of this membrane. Basically, what we believe is that there is something going on like membrane rupture, where membranes, heterogeneous membranes, we know that they are derived from the endoplasmic reticulum, rupture open, they are shaped to become to bend in a certain direction via the scaffold protein that associates with them. And then the crescent starts growing around these core proteins here in the middle of the virus. And basically, as the crescent membrane grows, more and more of these open membranes contribute to the membrane growth of it, such that eventually it's entirely spherical. And then, as long as the viral DNA is not inside this sphere, we know that there is an opening here, which we have observed by electron microscopy. And this opening is basically the opening through which the viral DNA is inserted into the particle. We have observed this process by tomography. We have seen how the virus is actually inserted through this hole into the immature particles. And then as soon as the DNA is inside, we observe that actually all those particles that have viral DNA have a closed membrane. So you see there is something very hypothetical going on here, and it is basically depicted here by these black dots. We think that because we would imagine that open membranes are inherently unstable in the cytoplasm, we basically have, in this model we hypothesize about some kind of complex, perhaps mediated by lipids, specialized lipids or proteins, that keep the membranes open as long as they have to be open, like here and here. But then at some stage there is some signal, probably mediated via insertion of viral DNA, that destabilizes this complex, and then basically the membrane can close. Now, of course, we would like to know what are these black dots. We have no idea. I will show you what they are not. What we try to do is basically naively to analyze mutants of the virus that are unable to make crescent membranes, assuming that these could give us a hint as what is required for membrane rupture. And all that we know is what is not involved It's not the scaffold protein, because as I showed you before, when you treat the cells with rifampicin, you make membranes that are unstructured, and these membranes are open. It's not one of the major membrane proteins of the virus, and although the virus cannot make crescents when you basically delete this protein, you start making these typical onion-shaped membranes, and when you analyze those, you can clearly see that they have open ends. So basically these onions are open membrane sheets that start to stabilize themselves by curling into onions, because they cannot associate with the viral zone. So then we decided, okay, perhaps what we should do is just look how general is this mechanism. Do other viruses also rupture membranes? And this brings me to this nuclear cytoplasmic large DNA viruses. And this is what I would call a cell biological view to these viruses. They share quite a number of common features. They have large genomes encoding for many proteins. They all replicate their DNA in the cytoplasm, which is quite unique. And they do so by forming large cytoplasmic DNA replication sites as well as assembly sites. Now, as a cell biologist, what I find very interesting is that they do so, and this is from our own work, by enclosing these DNA replication sites by the endoplasmic reticulum, forming what is called like a cytoplasmic mini nucleus. Some people believe that perhaps this was the origin of the nucleus. I think that this basically just represents a way of the ER, the dynamics of the ER. So we have observed this for Vaccinia virus. I recently observed this myself for another large DNA virus, African swine fever virus. And I was recently in a meeting where Abram Linsky also proposed that for Mimi virus, the DNA replication site is surrounded by the endoplasmic reticulum. So now I feel like here, this German man who comes from the south of Germany drinking beer and goes to China, and then sees that actually people are not drinking beer in China, but they're doing weird things. They're eating with the sticks. Because I'm going to move into something I don't know anything about, which is viral evolution. And I've taken this from a review. Basically, when you compare the genome of all of these large DNA viruses, hints may suggest that they actually derive all from a common ancestor. What is actually very interesting is that most of these other large DNA viruses have an icosahedral form, whereas Vaccinia virus is brick-shaped. What is very interesting indeed is that they share quite a number of proteins that, in which they have homologies, obviously proteins that enable them to replicate their DNA in the cytoplasm and to carry out transcription in the cytoplasm, but also some proteins that might be involved in virion packaging and morphogenesis. So what we asked ourselves is basically, besides sharing proteins, could they also share a common way of membrane acquisition? And I showed you some data here on Vaccinia virus up here, and then recently we moved into this virus here, African swine fever virus. And this was based on a collaboration that we started with Maria Salas in Spain. So this is a typical view of African swine fever virus. So as I told you, an icosahedral kind of morphology. And again, the details here, a large DNA virus replicating its DNA in the cytoplasm, encoding for many proteins, and it's a member of this nuclear cytoplasmic large DNA viruses. Now, it shares many parallels to pox viruses. I showed you one already, the DNA replication site being surrounded by the endoplasmic reticulum. But also, when you look at the literature, there's some interesting similarities here. Because some groups claim that the virus has a single lipid bilayer, whereas other groups believe that the membrane of the virus is derived from the cisterna, derived from the endoplasmic reticulum, which is collapsed. So, if you ask me, this sounds very much like Vaccinia virus, where one group suggests this model, whereas the other group suggests this model. It's just that we are dealing here with a different shape.